Hello and welcome. Today I'm going to talk about plant-based vaccines. During this discussion, I'm going to elaborate on what is the role of vaccines in human health. What are the types of vaccines available? And what is the role of plants in modern vaccinology? So, before I proceed, a request. If you have not subscribed my channel, please do so. And also hit the bell icon so that you get information about my future videos. So, what are vaccines and what is the role of vaccines to human health? You see, vaccines protect us from infectious diseases. And if you recall, entire population globally is vaccinated soon after the birth to prepare us to fight against the well-known infectious pathogens. In addition, if as and when a population is threatened by new pathogens, infectious pathogens, efforts are made to develop vaccines then and there or as soon as it is possible and use them to protect populations. Example, very recently we had this COVID pandemic came 2 years ago. Quickly, globally efforts were made and vaccines were developed and used. As a consequence, we could overcome this threat in the quickest time possible. Okay, so this is the role which vaccines play to protect human life against infectious pathogens. It's a well-known fact now. Now, if we see what are the types of vaccines available, okay, so we have conventional vaccines, and the new generation vaccines. Let us look at what are conventional vaccines. In conventional vaccines, whole organism is used and administered to populations or humans to induce immune response, to prepare them for fight against the disease. When we say whole organism, actually it is attenuated or heat killed or killed viruses or bacteria which are used. But this conventional way of making vaccine has a small risk. That is, they may get, though at a very, very low possibilities, but the organism can get activated. And when it gets activated, it can cause diseases. So that risk is there. I'm not saying it is happening. It happens in a very, very low proportion, may or may not happen, but that risk is there. And therefore, to overcome this risk, in recent times, new generation vaccines were envisaged, thought about based on our knowledge in the field of molecular biology, cell biology, immunology, that instead of using whole organism, we use the the specific gene protein or specific protein coded by a gene which is antigenic. which is actually responsible for pathogenesis. Okay, so we can, or we have now the protein-based vaccines or the nucleic acid-based vaccines. Okay, so protein-based vaccines are basically a specific protein which has been identified from the pathogen that this is the one which is responsible for causing disease, or it is the one which interacts with the host system. So instead of using whole organism, it was envisaged that we take this protein alone. Okay, and not only protein, the gene sequence or the gene which is coding for this protein was also used as nucleic acid vaccines, or the even mRNA which codes for this specific protein is also used for making vaccine. Examples, if you ask me, very recent again, during the last pandemic, we had vaccines available, as I mentioned earlier, and all the three types of vaccines were there. The vaccines made by conventional approach, taking attenuated virus itself, and also we had recombinant protein based vaccines, and also mRNA based vaccines which were developed and used. For example, in India, if you see, we used Covaxin, which is the conventional vaccine, and Covishield, which is the recombinant protein based vaccine, you know. And I'm going to actually elaborate this part towards the end of my session. So this protein-based vaccines, which I'm going to discuss slightly in detail, are also called subunit vaccines. Okay, now subunit vaccines, what is required here? You need to have a protein identified, which is pathogenic in nature, or which is the one which is responsible for causing infections and diseases, you know. So you once you identified a protein or a gene, it is the and not only the protein, you can also identify the part of the protein which is antigenic, what we call epitopic regions of the protein, which interacts with the host cells, you know. So not only one can use whole protein, in fact, you can use only part of this protein, which is the epitopic region, which is the immunogenic region, you know, and use it as vaccine, you know. Okay, so with this introduction, we come to the today's question. What is the role of plants in this whole process? Actually, plants have been used in two ways. One is a concept which started in mid 90s, mid 1980s of last century, that we make this immunogenic protein in plants, and the plants which are eatable, which we can eat. So the concept was to make vaccine in plants which are eatable. So the concept was known as eatable vaccines. Okay. Now, this had an added advantage over the conventional vaccine or vaccination or vaccinology. That is, the problems related to storage, transportation, which required very specific transport conditions and the storage conditions for the conventional vaccines is taken care of by plant-based vaccines. They're easy to carry. You can carry them under normal conditions because they are plants. And also, the conventional vaccines were administered through needles as injections, muscular application. And edible vaccine thought to have an advantage that you can just eat them, and probably they will do the job. So with this kind of concept, a lot of work was done in last 2 decades or over 2 decades. A lot of candidate vaccines were developed in plants, edible plants, but they could not be marketed or commercialized for human usage. I'm not going to talk about this aspect of plant usage for vaccines, but I can mention that why that failed or that could not take off is mainly because of the regulatory reasons that they required. The required regulatory regime is not available globally, which is acceptable, which will give clearance to these products, etc., etc. So, that approach remains on shelf. Though a lot of candidate vaccine were developed and they are waiting for clearances, but then what I'm going to talk about the other application of plants in vaccine or vaccinology. That is, plants are used as manufacturers of this protein-based vaccine or subunit vaccines. That is, once you have identified a gene or a protein which is antigenic, which causes disease, and also you have identified the epitopic region, which is part of what we call drug designing, then you can clone it in appropriate vectors and express it in plants. And now the whole thing will be amplified in terms of production. The plants are going to make this recombinant part of the protein, which is antigenic. Okay, and then you can purify this from plants and use it as vaccine. So that is the second application of plants and vaccines, that plants are used as manufacturers of a designed drug. Okay, or you can say, in other words, it is example of biofarming. So in a couple of slides, I'm going to explain to you what this biofarming or manufacturing of vaccine by plant is. And towards the end, I will tell you a success story about a vaccine which is manufactured by plants. Okay, so when we talk of plant making these vaccines or amplifying these the vaccines, actually there are two methods by which have been utilized. One is you can have nuclear transformation done to the plants, and you have transgenic plants expressing this particular recombinant protein, and making, and these plants are going to make this protein, and we can purify the protein or the vaccine from these plants. And the second approach is the manipulation of genome of a plant pathogenic virus. Okay, now this is going to have the transient expression of the recombinant gene in the plant cells. So it means each time you want to make a vaccine in plants, you have to infect the plant with that recombinant virus particle. Okay, I will tell you what these recombinant viral particles in a while. So these are two approaches available to make vaccine in plants. One is via nuclear transformations, and another by having engineering the pathogenic viruses of plants, infecting the plants. And as you know by nature, that once the any living organism, higher organism, gets infected by virus, it spreads from the site of infection to the entire plant body or animal body, So both the approaches were used, but I'm going to focus on how the pathogenic viruses were exploited to make larger quantities of vaccines in or subunit vaccines in plants. So as a requirement, as I was mentioning, that the requirement of a subunit vaccine is to have the suitable vaccine gene or pathogen, you know. And here you will see a list of genes, which have already been identified against the diseases they are involved in, So it means we have the candidate gene or protein in hand already being done. I mean, this work is being done. I mean, it requires hardcore molecular biology. So all these genes have been identified, including a spike protein gene from COVID-19. So this is the causative protein which was identified, from COVID-19, which causes the infection. So this is one part of the preparation or recombining. If you have a gene, you can try to identify, from the antigenic sequences, you can identify the epitopic region, which part of the protein is actually responsible for antigenicity or pathogenicity. You can amplify those regions from the gene sequences or the genome of the virus here, and then these are cloned into specific vectors, expressing vectors, and then this whole thing can be pushed into microbes if you want to make it in microbes or animals or plants, as the case may be. So you have to clone these regions which are coding for a specific epitopic region or antigenic region, which is responsible for pathogenicity in a vector, which should have or which should be appropriate depending on where you want to amplify these virus particles, either in a microbial cell, animal cell, or a plant cell. Once you have generated large amounts of this protein purified from either microbes, animals, or plants, they need to be checked in animal systems for their immunogenicity and then the efficacy of these vaccines of these subunit vaccines against the protection from the known pathogen, whomsoever. So this is the general scheme of things, from gene to protein, and from protein to the specific epitopic region, which is immunogenic, and then use that as as a recombinant vehicle to express that in a either microbial cell, animal cell, or plant cell. As the case may be, after amplification or making it in large quantities, you have to purify. That is, to keep this in mind, that purification of virus particles, recombinant virus particles, are very, very essential. And then that is used for their tests against the pathogen, in model systems, first in animals, and then, of course, finally in the humans as clinical trials. So this is the general outline. Now, once you have done that, you have identified a specific protein and part of protein, then you can express, or one can express transiently in plants via plant pathogenic viruses, by engineering that viral genome which you want to use. So what we are doing, we are taking that recombinant protein which we designed earlier and put it into plant pathogenic viruses. And then this recombinant virus is used for infection or infecting the host plant, you know. And then that host plant, when virus spreads, so these recombinant viral particles are made all through the plants in larger quantities. So the advantage of using transient expression of viruses is virus particles in plants is that in one go you get large quantities of these virus particles made in plants. And then you can purify these particles from here and use them as vaccines. So, for example, what is happening here? If it is a plant virus, and if you have engineered the epitopic region of a viral coat protein gene, and then it means you have created a chimeric gene of the viral capsid protein or coat protein, and then it is used for infecting a plant, and then you see the variegated region of plant leaves is because of the infection caused by this virus, And when you isolate virus from these plants and look at how the virus particle looks like, so you can see this is electron micrograph. Here you see the green and maroon are the native host viral proteins, and what you see here, a yellow colored thing, is the protein which is recombined in the viral genome or viral code protein gene, actually. So that's how they protrude outside. Since they are in code protein, and that region which protruded out has been engineered of the original code protein, So that's how it looks like. I mean, the viruses which are engineered using the gene of interest, or the epitopic region of a pathogenic protein from the virus. And this is just illustration of that, what I was trying to say. This is the healthy leaf, and this is the infected leaf. Virus is isolated and slightly closer row. So you see these yellow portions which are protruding basically is the protein which one wants to express on virus particles outside, so that they are exposed and can interact with the host. So basically, what is happening, to conclude here, that we try to generate virus nanoparticles, or in short, you can say VLPs actually, or virus-like particles, So this is normal protein, code protein like this, which get assembled into a virus. And then you have this chimeric VLP or chimeric protein, viral protein, wherein, this region has been replaced, or we have inserted the epitopic part of the antigenic protein from the disease-causing virus here, and it gets expressed like this. And when these particles assemble in the form of a virus particle, you can see these things are exposed outside. And of course, inside there will be the genome of the virus, like either DNA or the RNA. And there are other ways of having these engineered virus particles, like antigen display particles or RICs. But I'm going to focus on the VLPs, because that's where the success story lies. Although all the three types of viral nanoparticles have been generated for different vaccines, So, and if we see in last 5 years, a lot of work has been done. I mean, it is being done last almost a decade and a half. But if I just put or summarize here the work done by uh, various laboratories globally in last 5 years, you see uh, a variety of viruses which cause human diseases have been have been investigated actually, and from this point of view, making subunit vaccines means their candidate genes have been identified, the epitopic regions have been identified, and they are cloned into appropriate plant pathogenic viruses, and the plants have been infected, and vaccines have been made. For example, if you see Zika, this exercise has been done against Zika virus, yellow fever virus, rotavirus, norovirus, virus, and malaria, for example, anthrax, and HPV and influenza virus. So list is, I mean, almost all known human pathogens have been addressed uh, by this approach to make vaccines, okay, uh, including the SARS-CoV-2 vaccines for this COVID-19 virus, And here also, this VLPs, have been generated basically against the candidate proteins, including a spike protein, and they were amplified in a host like Nicotiana, Bendemiana. This is the host plant where, in fact, all these studies have been done. The epitopic region from different candidate genes of these of these viruses were identified and cloned, and this vaccine is made, or candidate vaccine were produced in tobacco plant, and then purified and tested for their immunogenicity in animal model system. And only very few have gone to the human trial level. That too, at the approval level, the studies must be going on to see the efficacy in human beings. So when we talk of this approach, very recently a success story came where we have first plant-produced vaccine for human use against COVID-19 was recently authorized by Health Canada after the successful outcome of phase 3 clinical trials. Now, this is the first example where a vaccine is made in plants, tested in human, up to human being in clinical trials, and it is cleared to be used by humans in Canada. Okay. Now I will elaborate a little bit here. So who did this? So this is the company who did, Medicago Inc. Canada. That is the company who made this possible to make the vaccine for COVID against COVID-19 by plants, So here they developed this vaccine using this spike glycoprotein or spike glycoprotein from this strain of SARS-CoV-2. So this is the strain they use to isolate this gene, spike glycoprotein gene, okay? And then identified that region and then cloned into a recombined virus. So what they did here, they took the influenza virus, the transmembrane region and the cytosolic region, and then attached to this the spike protein region of the CO2 or COVID-19 virus. So basically, it is a recombined protein between influenza and COVID-19, having the transmembrane region and the inside region from cytoplasmic tail region from the influenza virus, and the outside region is the spike protein region. Okay, and then they increase the VLP assemblies and budding. So uh, so they also did some manipulations in the spike protein region, where they tried to stabilize this cleavage point between S1 and S2 by making certain substitutions at this part, so that when this protein is dissociated itself, it is stable and it doesn't get denatured or changes after it is detached from the virus particles. So so this is what this was done scientifically, and then it went through the clinical trials. This co-VLPs or coronavirus-like particles, which were purified from the plants, were used into the phase 1, 2, 3 clinical trials to assess their immunogenicity, safety, efficacy, etc. And the encouraging results which were seen here is that they had the 78.8% efficacy against moderate to severe disease and a 74% efficacy against seronegative participants. Okay, so it protected to a larger extent here against the COVID infections in the trial experiments. Not only that, this was also effective actually against four different strains which were which were infecting the global population and created havoc, like Delta, Gamma, Alpha, Lambda, and Mu. So this vaccine was found to be effective against five different strains of the COVID-19. Okay, and finally, this was officially approved for humans or human use in Canada by their regulatory agency, Health Canada, and is now being marketed by Medicago Inc. with the name of Covifense. Covifense is the name given to this vaccine, which is plant-based, which is sort of manufactured by plants or amplified by plants, plant mechanism, and purified from them and used. So, so I stop here, and I have tried to give you an idea about vaccines in general, plant vaccines in particular, how plants have contributed or are going to contribute in the area of vaccinology. So plants can be used as sort of manufacturers once you have engineered and you have the gene which is responsible and identified the epitopic region, and you have cloned in an appropriate vectors, then the amplification part or the manufacturing part in larger scales is done by plants, which is safe, and I think it is economical as well by any other means. If you can do it in microbes, you can do it in animal cells as well, or in animals, but option of plants comes pretty handy. So thank you very much for patient hearing. And in the end, I would like to request again for request to subscribe this channel and hit the bell icon. See you soon.